Good morning. Thanks for being here. Functional neuroimaging evidence from the late 90s and more formally in the early 2000s and mid-2000s established the existence of these distinct large-scale networks in the brain thought to serve functions that have been referred to as external and internal attention. So the default mode network is deactivated during a wide variety of task conditions involving attention to the external environment, and conversely it's activated when we draw from internally generated thoughts, whereas the dorsal attention network and the salience networks are each activated in unique manners during tasks involving attention to the external environment. And beyond these engagements of these networks with task performance, a key feature of the networks is this within-network correlated activity, or also known as functional connectivity, during wakeful rest spontaneously, as well as during other states. And this kind of suggests that there's an intrinsic, this is an intrinsic network. So beyond the within-network connectivity, another sort of striking finding that's emerged from resting state fMRI is this idea of between-network negatively correlated activity or anti-correlated activity. And however, this still remains a controversial finding. And if you attended the workshop, symposium a couple of days ago called "The Global Signal Strikes Back," you would see that. This is a potentially important aspect of brain organization in that if there is an intrinsic negative correlation between these areas, it could suggest that these are areas, networks that are in continuous competition with one another, potentially for access to the same computational resources. However, we understand very little about the neurophysiological dynamics of activity within and between these networks on time scales faster than fMRI can measure. So I'm going to talk to you this morning about our work with human intracranial EEG to address this. And this work is done with patients with focal epilepsy who are undergoing invasive monitoring for seizure activity, either using the ECOG method of subdural grids and strips implanted on the surface of the brain, or the stereo EEG method of depth electrodes implanted in deep brain structures. They also traverse through areas on the cortical surface. And so the electrode placement is based on clinical need, and coverage isn't always found within and across major nodes of the networks that I described. But today I'm going to talk to you about data from particular cases where we did have simultaneous coverage and we can study these networks. So I'm going to break up the rest of the talk into two parts. So I'm going to tell you about first intracranial electrophysiology of within-network intrinsic functional connectivity, and then I'm going to use a similar conceptual approach to tell you about extending this to understanding inter-network interactions across various states. So in intracranial EEG, we get this super rich raw signal, and we typically perform time-frequency decomposition to break it down into distinct frequency ranges, ranging from delta, or 1 hertz, all the way up to gamma, or 40 to 70 hertz. And these are frequency ranges that are a common focus also in scalp EEG and MEG, and other speakers in the symposium will tell you more about network interactions within these various frequency bands. I'm actually going to focus mainly on a higher and broader frequency range that we call high frequency broadband, or HFB, that's 70 to 170 hertz, also sometimes called high gamma. And the HFB signal is a well-known correlate of local bold activity, or the fMRI activity, and it's also a correlate of neuronal population spiking near the location of the electrode. And with intracranial EEG, we get unique access to activity within this frequency range with high signal to noise. And a focus on this high frequency range has been fruitful in linking connectivity in electrophysiology with fMRI, and a key finding, a sort of seminal study done by Yuval Nir and colleagues, looked at this by looking at electrophysiological interactions between right and left homotopic regions in auditory cortex, where they showed that if you filter the high-frequency broadband envelope to the infraslow range, or less than 0.1 hertz, you see this tight coupling between left and right hemispheres during wakeful rest. And this is sort of analogous to the early Biswell and colleagues' finding of homotopic connectivity in bold between right and left hemispheres. And so, however, to really demonstrate that this is an intrinsic connectivity, it's important to demonstrate this in other states. And indeed, they showed that even during a sleep state, you see this very tight coupling between hemispheres. So we wanted to extend this kind of approach to look at the networks of interest like the default mode and dorsal attention network, and to link this directly with bold functional connectivity. So here I'm showing data from a patient who had simultaneous electrode coverage within the medial prefrontal cortex and posterior cingulate cortex, key nodes within the default mode network, and the patient underwent resting state fMRI prior to surgery. And the resting state fMRI showed that, as expected, there's this tight coupling of the bold signal between these two distant regions. I'm now going to plot the example correlation between these regions in the high-frequency broadband signal filtered to a lower frequency range. And indeed, in this 1 minute sample segment, we see tight coupling also in the ECOG recordings. So how anatomically specific is this? So these are distance regions from one another. So to do that, I'm plotting now a classical seed-based correlation analysis from a seed in the medial prefrontal cortex. On the surface of the brain, I'm showing the bold correlation with the seed, and on the electrode circled locations, I'm showing the ECOG correlation. And as you can see in both modalities, you see this hot spot in the posterior medial cortex with a nice correspondence between modalities, and indeed there's this tight spatial correlation between modalities within the same individual's brain. We're able to replicate this across various patients who had this unique coverage across the medial structures within the default mode network. And this also was found for the dorsal attention network. And so I'm showing 2 example patients here with nice fMRI versus ECOG overlap with, who had coverage in the frontal eye fields and dorsal posterior parietal cortex within the dorsal attention network. We've also looked at this during sleep. Here I'm showing resting state ECOG connectivity from the same seed region in 2 independent resting state runs, and you see a similar pattern in each run, so this it's reproducible. And now I'm going to plot that during sleep session, 4 minutes long. And here we see, again, we see this nice anatomically selective hotspot in the posterior medial cortex. And so even during sleep, we see this super tight coupling between these distant regions. Okay, so now that I've established kind of a way to, and a framework to look at interactions within these nodes of these major networks with fMRI, I'm now going to extend this approach to talking about internetwork interactions across various states. And I want to segue by highlighting this recent study that was led by Omri Raka. And he showed that he studied patients with simultaneous coverage within a major node of the DAN and the DMN. And I just want to focus on the red and blue curves on that plot. He showed that during a cognitive task involving mathematical cognition, following trial onset, the dorsal attention network node showed activation, shown in red, and the default mode area showed deactivation, shown in blue. And as you can see, there's this clear temporal offset between the two by several hundred milliseconds. And so that's something that fMRI is blind to. So we wanted to extend this to looking at deeper questions like how do nodes of the DMN DAN as well as the Salience network interact during task, rest, and sleep. And we asked whether do fast internetwork dynamics vary with cognitive state. And to engage these networks, we had patients perform tasks used in previous fMRI studies known as the gradual continuous performance task that's known to dissociate the functional profiles of these 3 different networks. And so in the task, the patient has to press a button for every city that's seen, and withhold a response for the more rare target stimuli, which are the mountains. So they're pressing a button for every city stimulus and withholding a response for the mountains. It's kind of like a go-no-go style task with gradually changing stimuli. And so these mountain stimuli are highly behaviorally relevant and are salient. And so I'm first going to focus on the response of major nodes of these 3 different networks to those mountain stimuli. So here's an electrode within a sample patient within the dorsal posterior parietal cortex within the DAN. And following the target onset, we see both for correct trials and error trials an increase several hundred milliseconds later. There's this very transient increase following the onset of the mountain. I'm now going to go to an electrode in the posterior cortex within the default mode network, and we see this high-frequency broadband decrease, which is very analogous to deactivation that's seen in fMRI, both for correct and error trials. Now I'm going to go to the dorsal anterior insular cortex, which is part of the Salience network. And we see this increase for both error and correct trials, but with a greater increase for errors and more prolonged increase. And this is a well-known feature of areas of the Salience network showing this error monitoring type signal. But now with the direct recordings, and we have access to fine-grained temporal information within and between these networks. And I just want to highlight the anatomical selectivity of these kinds of responses. So here I'm showing a patient who had a subdural grid on the lateral surface, covering areas of the default mode network shown in blue and areas of the DAN shown in green. And I'm going to zoom into 3 example electrodes in the lateral parietal lobe, with 2 in the DAN and 1 closer to the DMN. And we see these highly divergent responses to those mountain target stimuli, even though these electrodes are just millimeters away from one another. So this is clearly highly anatomically selective and is not really related to volume conduction. With the simultaneous recordings across these networks, we can now look at the interactions across various states and look at this idea of intrinsic anti-correlation. So to do that, we took these functionally localized, based on the task, electrodes within the posterior cortex and in the dorsal posterior parietal cortex, and we filtered the high-frequency broadband envelope to the infraslow range, more analogous to the range that's seen in fMRI. And across various states, we looked at 100-second independent time windows in task, rest, and sleep, but with about half an hour of data in each state in 3 different patients for these particular nodes of these networks. And what I'm plotting here is now the correlation between these areas in task, rest, and sleep. So in 3 different patients here, what we can see consistently is that there's an anti-correlation that's consistent across windows during task, but it's not seen as consistently during rest and sleep. However, the brain seems to visit this anti-correlated state intermittently during rest and sleep. So that's for the posterior cortex and the dorsal posterior parietal cortex, and now I'm going to plot that for the PMC versus the dorsal anterior insula and the Salience network, and it's a very similar pattern. So anti-correlation is seen consistently during task, but not during rest and sleep, and we get this intermittent anti-correlation. So we wanted to better understand what could be driving this variability in rest and sleep states, and we hypothesized that these anti-correlated windows would be associated with an overall brain state that's more similar to a task-like pattern. So to test this, we looked at, we took all channels that, or all electrodes that were implanted within a given subject's brain, and we calculated an all-to-all connectivity matrix in the task. So we created this task template shown in the middle, and then for different windows during rest that were classified as high anti-correlation or low anti-correlation or absent anti-correlation, we looked at the similarity of these matrices. And what we found was that when there was more anti-correlation, the brain state was more similar in the topographic pattern to the task-like state. That was seen for anti-correlated states in rest compared to task, and it was also seen for sleep compared to task. So even during sleep, when anti-correlated states were visited, the organization was more similar to task. And if this signifies a more vigilant, attentive state that's visited, then we would also expect that task performance would increase when you have a more anti-correlated interaction between networks. And to look at that, we looked at the variation of these anti-correlations across various sessions of task performance. And so here I'm plotting the high-frequency broadband envelope that's filtered to a lower frequency range. And we looked at the zero lag correlation between the areas, which is analogous to what's done in fMRI functional connectivity. But in addition, because we know from Omri's work that there's a lag between these networks, we also looked at this lag minimum correlation value with shifted correlations between these areas, with the dorsal, the DAN leading the DMN in this particular example. And we hypothesized that this lag minimum correlation value, when it's greater, would be related to greater task performance. And so across these 6 different patients that I'm showing here in different colors, we see this negative correlation that shows that when there is greater lag minimum correlation between the DAN and DMN, there's better task performance, as indicated by D prime, which is a metric that accounts for false alarm and hit rate in the task. However, when we look at the zero lag correlation, we don't see this relationship, and we establish this more formally with a linear mix model. So we only see this brain behavior relationship when we account for this lag between the networks, and it indicates that a more anti-correlated state is indeed linked with greater task performance. So to summarize, I've told you that within network intracranial EEG, high-frequency broadband connectivity, including within these major networks, the default mode and dorsal attention network, spatially matches bold topographic patterns. Between network, infraslow anti-correlated activity is found reliably during tasks, but emerges only intermittently during rest and sleep. And these activations in the DAN and SN precede DMN deactivations by several hundred milliseconds, and greater lagged anti-correlation between the DMN and the DAN is associated with better attentional performance. So I hope that these findings are taken into consideration when interpreting fMRI functional connectivity findings within in between networks and in healthy and disease populations that many people here study. So thanks so much for your attention. I look forward to the discussion after.